I'm going to try my best. There's going to be a lot of formulas on the slides, but it's not necessary that you understand all of them. I'll try my best to give you kind of the high level overview. So just really quick to introduce myself, my name is Lisa Bramer. I've been at PNL just about 10 years now. I sit in the computational biology group and lead a team of biostatisticians and data scientists. And my email is up here. If you have questions afterwards, I'm happy to field those. So we'll leave off where Rachel was talking. We have, right, your crosstab that has genes as your rows, samples as your columns, and your entries are counts. And hopefully you've done some sort of quality control, pre-processing. And then we're ready to say, well, okay, can I actually analyze my data and say anything about whatever it is that I'm studying? So after you've cross-tabulated these, you've done some pre-processing, and then we want to do some modeling. And what people in the field refer to this as is differential expression analysis. Okay, people often come into this step and they say, oh, good, I'm finally going to start to get some answers. This is one of my favorite cartoons. Like, as soon as you start analyzing data, you might create more questions than answers. So that's something to be aware of. Right, so both Bill and Rachel talked about this some, but one of the primary things, right, that we need to consider is this library size. So if I have two samples, two different libraries, and we see that one of them has a total of 10 million reads, the other has a total of 5 million reads, I'm going to expect by random chance that, I'm going to expect that overall for each gene, I would expect a twofold change if counts were just randomly distributed amongst the genes. So this is the slide you've already seen from Rachel. There are a bunch of different ways of trying to deal with this. And we've already kind of gone through this in painstaking detail, right? The thing that I want to say about this is that a lot of the numbers that are in here where they're adding a half or 2, these are very arbitrary. And Kelly talked a lot yesterday about statistical analysis of proteomics data. That field is 10 to 20 years ahead of transcriptomics. It is still the wild, wild west when it comes to statistically analyzing transcriptomics data. So I would just caution you to make sure that you are looking in multiple places or talking to statisticians or multiple people when deciding how to analyze your data, because, well, we all know not everything you find on the internet is true, but in the case of this type of data, people are doing all sorts of things and just kind of making it up as they go. So when we talk about scaling or normalizing transcriptomics data, we can also talk about this trimmed mean. This is another potential way that we can think about normalizing. So here we're looking at a log2 where we have a count for a given gene and a given library or sample divided by the total count in the numerator. And then if we have some sort of reference library, then we can take the count for that divided by the total counts in your reference library. So basically, you're normalizing to a reference library. In this case would be a single sample that you trust more than any of the other samples for some reason. So sometimes in experimental designs, people will choose like the control samples to be kind of their reference library to normalize everyone to. The other thing that people do, which we haven't talked a ton about, is if you haven't noticed a theme this week, the theme of a lot of biological statistics is trying to manipulate data so that it approximately follows a normal distribution, because a normal distribution has nice properties. They're the types of models that have been around for over 100 years. So in the transcriptomics case, that is what lemma-voom is attempting to do, or lemma paired with voom. That TMM normalization that I just showed is also attempting to do that. And there's a third method that is based on what's called compositional analysis. So, and that common package people use for this is LDEX2. And I'll talk about when you might want to use this approach versus some of the other approaches in a minute here. But the compositional approach basically comes from thinking about fractions of the whole. So back in the days where they used to mine for ore, they would think about things about what percentage of my stuff that I dug up is gold, what percentage is whichever ore elements that they cared about. And people have taken this and tried to extend it to things like transcriptomics or 16S data. Okay, so if we talk about differential expression analysis, and we're not in the case where we're trying to make things look like a normal distribution, back at the very beginning of this field, people were really trying to make things fit a Poisson distribution. I promised you that you don't really have to care that much about the equations that are happening here. But the traditional sort of interpretation of a Poisson distribution is some number of events that occur in some fixed interval or time or space. So we could think about the number of times you see a gene in some fixed sample. And that's how people started thinking about, maybe this follows a Poisson distribution, these counts for each gene across a bunch of samples. One key thing about the Poisson distribution, at the very bottom here, we have the mean of the Poisson distribution is this Greek letter lambda. And by definition, the variance of that distribution is also lambda. So the mean and variance have to be equal with this distribution. And as people started to use this model, you could see what we can do with the Poisson distribution. So at different values of lambda, so you can see that at lambda equals 1, I get lower levels of counts if I just simulate data from that distribution. At lambda equals 3, the counts start to look greater. At lambda at 5, the middle of the distribution increases, and what you start to see as lambda increases is that the variability or the spread of these distributions also increases. And that's because of this assumption that the mean is equal to the variance. So a lot of early RNA-seq studies were based on technical replicates rather than biological replicates. So in other words, taking samples or taking a sample and prepping it into multiple aliquots and running it. Just, I mean, it makes sense when a technology is new, you want to show that basically you have reproducible work. And in this case, a Poisson distribution was actually not a bad approximation for any given gene and the counts that you would observe. However, now that we've moved on to something more complicated where we're actually looking at biological replicates, what I have here on the x-axis is, so for a transcriptomics data set, for each gene, we can calculate the log mean count. And this is, the log mean count where we've added some small number to everybody, so we're not taking the log of 0. We can also, with adding a very small number to all our counts, compute the log variance of each gene. So if we were still talking about something that could be assumed to follow a Poisson distribution, it should follow this straight pink line. This is just a standard transcriptomics data set like Rachel showed. The color of these bins is the number of genes that sort of fall into that bin. And what we can say is that these data, a Poisson distribution does not approximate this blue curve that's showing you what the trend in the data actually is. So with modern day transcriptomics experiments where you're doing multiple biological replicates rather than technical replicates, a Poisson distribution, this assumption that the mean is equal to the variance turns out to be a very bad one. And the field so then moved away from using this distribution. When data do not follow this straight x equals y line, this is you'll hear people say a term called over dispersion, which just basically means that your variance is greater than your mean. Right? And so we need to think about a different distribution instead. And the higher your counts for your transcript or your gene, the more we tend to really violate this assumption. And so if you think about that, that's pretty disconcerting, since most of the time when we run statistical analysis, the things that are higher in counts or abundances are the things that we usually have the best shot at finding to be significantly different between biological replicates. And we're in a case where we clearly are violating the assumptions of the model there. All right, so instead of a Poisson distribution, data scientists and statisticians turn to a negative binomial distribution. And so this is similar to a Poisson distribution, right? Where it's what we call a discrete distribution, meaning the outcome is sort of a countable set, or if you think about transcripts, they are literal counts. And if you talk to a statistician, the general interpretation here is the number of successes in a bunch of independent trials before you reach some number of failures. That does not translate very well. Like when I was talking about the Poisson, we said, well, we can think about it's the total number of occurrences in some finite space. We could think about that making sense. This interpretation doesn't necessarily translate super well, and is part of my argument for why sometimes we're going to talk about this model and how people have formulated it, but it sometimes is not the best approach. But the benefit of this model is we're still assuming data, like for a gene, to be counts, but now we get this additional variance parameter. So if I rewrite what a negative binomial distribution looks like, we can rewrite it in this form where mu is my mean number of counts and sigma squared is my variance. And now I have two different parameters for the mean and variance, so they do not have to be equal, which is the major benefit here. So now you can see if I simulate data from this distribution, I can simulate it with mu equals 1, and I can start to manipulate how much variability there actually is in the data that I'm simulating, unlike the Poisson where they have to be exactly equal. What I will say is the other problem that people don't talk about is this negative binomial distribution. One characteristic of transcriptomics data, particularly now with RNA-seq, is you can see a lot of zeros. So everything I'm going to talk about after this, I'm going to talk about the negative binomial distribution for some of these models, but these are really what people call zero inflated negative binomial distributions. So they're using this negative binomial distribution, but they're adding in some other details that sort of allow for where you might see more zeros than you would expect with this negative binomial distribution. Okay, one problem with this model is the negative binomial, and then you add this zero inflation, is a very complex model. If you go to any of like the leading papers that implement these types of things, you start to look at equations. Even as a PhD statistician, I look and I'm like, what are you guys doing? Like, there's so many parameters. So one of the key problems I have is that we talked about that the mean is no longer proportional to the variance. So rather than just assuming that we know what that is, we now have to estimate the variability for every gene. And most of these methods, because RNA-seq studies, our RNA-seq data is very expensive, right? We tend to try to borrow information about the variants from all the other genes in your data set. And this will become important when we start to look at some variance plots, but the commonly used modeling approaches that kind of use this zero inflated negative binomial distribution are EdgeR or DESeq2. Those are by far and away the two most popular packages that get used. So let's just briefly talk about what happens. I'm going to go through this with how DESeq2 works. We will not get into the full mathematical details, but kind of the key steps, and then I'll tell you how EdgeR differs. So DESeq2, assume the key thing here to keep in mind, the assumption is that most of your genes are not going to be differentially expressed. So if you are studying samples that you know are going to be vastly different, this is probably not the analysis model for you. Right? If you think, a majority or even a large proportion of your genes should be differentially expressed because you're studying different biological conditions or treatments that are just going to be very different, this is probably not the way to go. What they do, and I think Bill mentioned this too, is so both DESeq and EdgeR, when you go to run it, you just give it the raw count data. You do not need to do any sort of transformations. Those transformations are part of the model implementation and estimation. But basically what this thing is going to do is for each gene, it's going to compute some of these scaled normalized values. And that's basically your counts divided by what's called a geometric mean. And it will get log2 transformed. And, the details here again aren't like super important. We're just saying we're getting things on a log scale where variances can start to be equal or stabilize, and we're getting these into something where we're kind of accounting for library size. So that is the first step of DESeq2. Next, it tries to estimate dispersion or variance. Those are interchangeable terms here for each gene. And what it's doing is it's taking the mean count for that gene, the mean normalized count, plus some value alpha times your different gene counts squared. So they're still trying to make this a function of the mean, but it's no longer just that it has to be exactly equal to the mean. And they're going to try to estimate this value alpha for every single gene. When they do this, what ends up happening, if you look at this plot at the bottom, you've got the mean of sort of your normalized counts. Your dispersion estimate is your variability. And what ends up happening is that function that we saw on this previous slide of the mean plus alpha times the counts. That's the red line here. And they go in and they actually do a transformation again in order to try to shrink your variability within a gene to get closer to this red line, this function that it's making an assumption about. You don't need to legitimately, you don't need to understand what's happening here. The key here is for you to know that the data is being transformed so it can try to fit this red line, because when you're running the models, looking at this sort of fitted line versus your actual estimates is going to become a diagnostic for whether this is a good model or not. And then the last thing they do is they will fit a generalized linear model to each gene. So you have these normalized and variance shrunken values, right? And you make them a function of some experimental factors you care about. We assume that all your transformed data follow this negative binomial distribution that also includes a parameter to account for lots of zeros. They'll run what's called a likelihood ratio test. So you can think of like when you're doing normal stats with like a Ttest or something like that. Similar concept. And then the one thing I will say is when we do transcriptomics analysis, I think I wasn't here for all of Kelly's talk, but sometimes in those types of studies with proteomics, we have enough or not enough replicates that we won't always do a false discovery rate correction if it's an exploratory study. With transcriptomics, you're going to have thousands to tens of thousands to hundreds of thousands of genes, so it usually becomes very imperative that you do a false discovery correction on your p-values. Okay, how does EdgeR differ? I'm going to go through this fairly quickly, but so the way Rachel mentioned, the way that it does this initial scaling in the modeling, again, EdgeR is doing it in the background. You do not have to do this. You give it the raw counts. But the way they do the scaling is a little bit different. The way that they estimate variance or dispersion, it's very similar to DESeq2, but they use what's called a robust dispersion estimation function. And it should, in theory, for most data sets, reduce the effect of outliers. Okay. The other key thing to mention here is this EdgeR still operates under this assumption that most of your genes are not differentially expressed. And it's going to do the same thing as DESeq2. It is going to also use a negative linear model or generalized linear model with this negative binomial zero inflated distribution. So what are the primary differences between these two methods? They're fundamentally similar philosophies, but the way that they normalize counts is different. The way that they estimate that variance or dispersion parameter is different. And if you search literature, there have been lots of reviews of all the different methods to estimate transcript or to model transcriptomics data. The consensus generally is that DESeq2 sometimes tends to overestimate variability, and that often can lead to higher false discovery rates. DESeq2, if you don't turn it off, and we can talk about this afternoon too, but in trying to control for false discovery rate, there is sort of an automatic step where if you're not careful in R, that DESeq2 will actually just start filtering genes out to try to get the most number of significant transcripts while still maintaining a false discovery rate. As a statistician, this feels very hokey to me. Like they're just fishing and trying to get as many things as they want. They have some justification in their documentation, but the thing to be aware of is that this happens by default if you don't turn it off. And DESeq2 will flag genes that have large outliers or remove these outliers when if you have enough data. EdgeR, to me, the benefit is that it is more flexible, but it's less user-friendly when it comes to implementing. You really have to have a better understanding of what you're doing and what choices you're making and why. Okay, the primary caution areas here, I've alluded to one already, is that these methods assume that a majority of your genes are not differentially expressed. So if you have libraries or samples that are very, very different, this is not the method. These are not the methods for you. And then it's an area of caution, not necessarily a bad thing, but these rely on these negative binomial distributions and these dispersion estimates. And on the left is a diagnostic plot that will get generated by these packages. So the points here, again, this is the mean normalized count and the variability on the y-axis. And this red line is what, I think this is DESeq, what DESeq is trying to fit. And then you see all the points around it. And I hope you look at that and say, yikes, right? That does not at all look like it follows that line. Something on the right is starting to get a little bit better, but this is DC2 where it's flagging these blue points up here are all things that are just really far away from that fitted line. And they tend to worry about the cases where your variance is much systematically larger than the line than below. So they don't flag a lot of these things down here, but these also look like they could be problematic. Okay. The other philosophy is that we can have transformation-based methods. So one of them we mentioned was Lemma, the R package paired with Voom. And they do this transformation that Rachel showed you previously. Again, this is a package where you would just give the raw counts and this does this automatically. The difference is it does this transformation / normalization to your library size, avoiding zeros in numerators and denominators. And it also tries to adjust your data so that your sort of average abundance and your variability, it tries to estimate that trend and maybe do another transformation to make that fit. But at the end of the day, after it's sort of trying to shrink some variance, it actually is using an approximation of a normal distribution rather than a negative binomial distribution. Okay, last method that we'll talk about is this compositional approach, LDEX2. It does something very strange, but effective, which is that this epsilon, it adds a small number to every count in your data set. So for a given gene, you're going to add some small number epsilon to every count. You're going to divide by this nth root of the product of all of these things is a geometric mean, and you take the log of that. You'll often hear this referred to as a centered log ratio. And if you really dig into the documentation, there are other alternate denominators if you have some certain characteristics in your data. So if you look at the LDEX2 package, they tell you how to look for these things. But if, for example, you have one group of replicates that you're studying and they have far more zero counts than the other, you might switch out this denominator. The package itself, it also just wants the raw counts and it does this transformation for you. But what it actually does is it takes and it runs a Bayesian model. So if you're not familiar, a Bayesian model is going to assume some sort of prior distribution on your data. You collect the data, you update that prior distribution, and we can do this many times. So what they really do is they take random draws from, it's called a Dirichlet distribution, and they do this many times to do simulation. So if you've heard of things like multiple imputation or other methods that are using bootstrapping, it's a common thing to do in statistics. Knowing that if I take one random sample, right, I go take another one, it's going to be potentially completely or different. Once they've done that, they compute test statistics. And so this is sort of what it looks like in the steps. So they take, you've got for one gene here, your counts plus a prior, in this case, it's a half. They draw these counts from this Dirichlet distribution. So if they did this 3 times, you can see this would be your first random draw from the distribution, second and third. And then they do what's that centered log ratio transform to get something that at least starts to look like the log of these values. Usually they do this more than 3 times. They do it like 200 times. And when they've done this multiple times for every random draw, they then come in and they say this CLR transform data follows approximately a normal distribution. So I can move on with my life and do a standard like analysis of variance or Ttest, depending on how many groups I have. They derive a bunch of p-values, one for each random draw, do a false discovery rate adjustment, and then your final sort of p-value becomes the mean of your adjusted p-values across all of these draws. So the people who developed this package have a paper where they argue that this is like the approach. I think like anything, if you look across literature, you can use any model to find pretty much just about anything you want, right? So it's not a matter of any one of these models being ubiquitously better across the board. What it really is a matter of knowing the assumptions of the model and when it's most appropriate to use it. So the statistician's favorite answer, you're going to ask me which method is best. And every statistician's favorite answer is it depends. And that holds here. But what it mainly depends on is your experimental design and your data. So when you have your data, you're going to think about things. The assumptions for DESeq2 and EDGE-R, these are highly complex models. And like I mentioned at the beginning, they're sort of just wild, wild west. So you have very stringent assumptions. We looked at some of those variance versus mean plots. We talked about that most of your genes, you should be expecting a majority of them are not differentially expressed, right? But we have to evaluate those assumptions with every data set. Lemma with VUM or EDGE-R. So if you decide that you're in sort of the camp where maybe DESeq2 or EDGE-R are appropriate, you have things you don't expect them to have huge differences. You look at some of your diagnostic plots and things look okay in terms of variance estimation. We mentioned DESeq2 is probably more user-friendly. It does some weird things like these automatic filtering. You also can't compute more than one like A versus B comparison at a time. So if you want to compare, you have 3 groups and you want to do all pairwise comparisons, you have to run things 3 different times. Not only do you have to run it 3 different times, if you just run it once, it's going to be computationally more expensive and take longer than EDGE-R or this lemma with VUM. And the reason that's important is if you're studying soil, plants, things that have meta-transcriptomes, where you have, like Rachel and I were dealing with the other day, you have a data file that's 5 gigs. This starts to become really important. The methods that are more like lemma VUM and ALDEX, so a lot of literature says they do a really good job at being under the sort of whatever you set your false discovery rate at. So meaning you're not going to get a bunch of false discoveries. However, if you remember, I think Javi and Damon were talking about this, earlier in the week, if you remember, false discoveries, less false discoveries come at the price of less true discoveries too. So you really have to think about your application. If it's if this is an exploratory study where you just want to know what's potentially interesting, maybe those false discoveries don't really matter as much. But if it's a study where you're really trying to prioritize candidate biomarkers to go study further, you probably want to make sure that the candidate biomarkers you get are actually true candidates before you spend more money. I will say if you have very small replicate sizes like n equals 3 per group, you probably are just as good to do nothing with the data than run like ALDEX or lemma VUM. They don't have the same power to detect things like DESeq2 and EDGE-R, particularly if you have small changes. This graph is from a review paper where they compared a ton of these different methods. This is false discovery rate. This is just showing what I've already kind of said in words, which is this lemma and lemma with VUM, the solid blue, solid black, and are down at the bottom here in terms of false discoveries. and things like DE-seq, which is in green, and EDGE-R, which are blue dashed, are slightly above. So we just tend to see that they have higher false discovery rates. The typical results that you're going to see from this, this is leading into Jason, who's going to talk next. But the typical output types you might see at first glance from a statistician are things like volcano plots, where we look at the log fold change versus the negative log p-value. If you're doing something like soil data, this becomes basically just a Rorschach test, like just ink blots. Not helpful. Or they'll try to make a plot like this, which is the average log counts per million and the log fold change. Again, as soon as you get lots of transcripts, I don't really know what you do with this. That's when you go and talk to Jason, who's going to talk next, and tell you what to do with this. And people will often try to make heat maps as well. But again, to me, this is like wholly unsatisfying because I have no idea what gene is what and really what's going on. I can see high level trends, but that's about it. So really, we need to put things in perspective.